You may have heard before the famous phrase that goes, "Cells that fire together, wire together." This is a phrase by Hebb which introduces the synapse's central role in the learning processes in the brain. Although the idea of synaptic strengthening through a co-occurrent activation wasn't new, Hebb developed a more precise framework, positing that as the activation of one cell repeatedly activates another, the cells become associated in a way that activity in one facilitates activity in the others. This results in subnetworks, where activation of only part of the neurons in the network is sufficient to activate the remaining ones and complete the pattern. This resulted in Hebb's main theory, known as the cell assembly theory. This theory was later supported by several observations in animal models, such as aplysia. Aplysia is a slug-like invertebrate in which serotonin-mediated transmission caused enduring behavioral changes. Historically, the sea slug has been used to investigate memory and learning phenomena. One crucial observation consisted in poking the slug's siphon, which caused its gill to contract in response. This could be interpreted as some sort of a protective reflex due to induced fear. However, as poking went on, the response became fainter and fainter due to a phenomenon known as habituation. A subsequent manipulation therefore consisted in applying an aversive shock to the aplysia's tail, enhancing the gill contraction reaction. This enhancement of the motor response to siphon stimulation is called sensitization. When stimulating the aplysia's siphon, the first neurons to fire are sensory neurons. They are wired to interneurons, which they activate through glutamate-mediated synapses. The interneurons then in turn stimulate subsequent motor neurons, activating the gill contraction reflex. With time, this reaction is hindered by habituation, and identical sensory stimulation produces much weaker motor signal. Tail stimulation, through an aversive shock for instance, can however be used to enhance the protective gill contraction reflex. The sensory neurons coming from the tail also carry excitatory input to the modulatory interneurons. In turn, these interneurons have output axon-axonal synapses wired to the siphon sensory neurons. Stimulation of the tail therefore enhances the signal brought from the siphon, enabling a higher motor response. This enhancement is due to a release of serotonin by the modulatory interneuron in the presynaptic axon terminal of the siphon sensory neuron. The serotonin released by the modulatory interneuron is then captured, enabling the release of G proteins in the postsynaptic neuron. This entails a series of reactions in that neuron. This all ends with blocking of potassium channels on the presynaptic membrane, causing longer action potentials. The transmitted signal is therefore stronger, and so is the motor response. But the interesting thing, when talking about learning, is not only the sensitization effect, but also the fact that it can be an enduring effect. What was observed in aplysia is that as serotonin binding to the siphon sensory neuron can directly cause potassium channel blocking, can also, through repeated processes, cause the synthesis of proteins which enable the perpetuation of the phenomenon for several minutes. At that point, serotonin release is no longer necessary. The enhancement of the response has been learned. Indeed, Kendall observed that, as with human learning, memory storage for each type of learning in aplysia has two phases: a transient memory that lasts minutes, and an enduring memory that lasts days. Conversion from working memory to long-term memory storage requires spaced repetition. As Kendall himself stated, practice makes perfect, even in snails. A second experiment, dating back to 1973, provided complementary evidence from the rabbit hippocampal area. This experiment, carried out by Bliss and Lomo, relied on previous work regarding high-frequency potentiation of synapses. It showed that stimulation of presynaptic neurons in the entorhinal cortex induced activity in neighboring neurons from the dentate gyrus. This induced activity was all the stronger when stimulation was fast. However, these observations did not yet predict long-lasting effects, although they clearly illustrated the connectivity between neighboring regions. Some years later, Lomo and Bliss carried out a variation of the previous investigations. They first stimulated the neurons afferent to the dentate gyrus at a calm and regular pace. This elicited a response in the postsynaptic neurons at a baseline level. Acceleration of stimulation, that is, a high-frequency stimulation train, showed an increase or potentiation of the synaptic response. This response, measured as the excitatory postsynaptic potential, or EPSP slope increase, actually remained for hours after stimulation. This discovery paved the way for later investigations, which shed light on the link between long-term potentiation and lasting modification of synaptic strength and activity in a way allowing learning. So, how does the long-term potentiation work? When an excitatory synapse is activated, the presynaptic neuron releases glutamate neurotransmitters into the synaptic cleft. These molecules can then bind to the neuroreceptors on the postsynaptic neuron. The main neuroreceptors for glutamate are AMPA and NMDA, which are both ionotropic receptors. Initially, the NMDA receptors are blocked by a magnesium ion, so only the AMPA channels can let sodium ions in. This initial accumulation, however, when sufficiently strong, pushes out the magnesium ion and opens the NMDA channels. A series of subsequent reactions accelerate accumulation of sodium ions as well as enable inflow of calcium ions. More neuroreceptors accumulate on the postsynaptic membrane thanks to these ions, and the synapse ion transmission capacity gradually increases throughout repeated activation. As we have seen, although Hebb was not the first to posit the central role of synapses in learning processes, he formalized a major theory regarding the strengthening of connections and subsequent learning. This theory was then supported by findings in aplysia simple nervous systems, as well as in more complex rabbit hippocampal systems. It paved the way for subsequent research and advances on the topics of learning, memorization, and more specifically, pattern completion and invariance to changes in neural representations.